Welcome to Virology Conversations, where we break down complex topics into digestible discussions. We take carefully curated scientific sources on a virology topic and generate AI podcast-style conversations. Think of this as your companion for better understanding the most complex topics. In this conversation, we're exploring how influenza's constant antigenic evolution shapes vaccine design, and how focusing on conserved viral epitopes could transform future vaccine strategies.
Today, we're tackling a real puzzle, influenza.
Yep.
We've been vaccinating against it for nearly 70 years, spent billions, and yet, every year, like clockwork, the flu's back, needing a new shot.
It really is the ultimate moving target. That's the core challenge, isn't it? You'd think after, 7 decades, our whole approach would look radically different. But fundamentally, the principles stayed pretty much the same.
Because the virus just keeps changing.
Exactly. It mutates, it escapes our defenses, and we're stuck in this cycle, always trying to catch up.
Okay, so that's our mission for this deep dive, to unpack how the virus keeps moving, why that target shifts, and then look at the really cutting-edge stuff researchers are doing now. Not just catching up, but maybe, finally, getting ahead with a universal vaccine.
Sounds good.
Let's start with that evolution then. Our sources point to two main ways the virus messes with our vaccination efforts.
That's right. The first one, the most common, is called antigenic drift. It's more subtle. Think minor, gradual changes.
Like little typos accumulating.
Sort of, yeah. Small point mutations crop up in the viral genes, especially the ones coding for those key surface proteins everyone talks about.
And why so many mutations? Is the flu virus just sloppy when it copies itself?
That's exactly it. The enzyme it uses to copy its RNA genome, the RNA-dependent RNA polymerase, it's notoriously error-prone. It doesn't have the proof-reading tools most organisms use.
Ah, okay.
So these errors, these mutations, they just happen frequently. Season after season, they build up, leading to new strains that our old immunity doesn't recognize as well.
Yeah.
That's how older strains get replaced.
And I read that this happens at different speeds for different flu types.
It does. The H3N2 viruses are, infamous for this. They drift and turn over much faster than A, H1N1, or the influenza B viruses.
Making H3N2 the big headache for the vaccine committees each year.
Definitely. It's the one that keeps them up at night.
Okay, so that's drift the slow creep. What about the second mechanism, antigenic shift? How's that different?
Oh, shift is way more dramatic. If drift is editing the manuscript, shift is like throwing the whole thing out and swapping in a completely new one.
Wow.
It involves a complete exchange of the genes for the surface proteins, the hemagglutinin, HA, or the neuraminidase, NA, or sometimes both. It's a major overhaul of the virus's appearance to our immune system.
And this big swap, this shift, it only happens with influenza A viruses. Why just A?
Good question. It's because influenza A has these huge reservoirs in animals, especially wild water birds like ducks and geese.
Right.
These birds carry a massive variety of flu strains. Now, if a person, or maybe a pig, gets infected with a human flu strain and an avian flu strain at the same time.
They can mix.
Exactly. The flu virus genome is segmented, it's in pieces. Inside that doubly infected cell, the pieces from the human virus and the bird virus can get mixed up and repackaged together. This reassortment can create a brand new virus with, say, bird surface proteins on a human virus backbone.
And we'd have basically no immunity to that new combination.
Precisely. That's what leads to pandemics. The population has very little or sometimes zero protection. We saw that in 1918, 1957, 68, and most recently 2009 with the H1N1 swine flu.
So the constant threat of both drift and shift is why we have that huge global surveillance effort led by the WHO.
It's a massive undertaking, yeah. Scientists all over the world track how these viruses are evolving, trying to predict which strains will dominate the next season so they can pick the right antigens for the vaccine.
But there's a built-in problem, isn't there? The timing.
That's the unavoidable flaw. Our sources really emphasize this. It takes 6 months, sometimes more, to actually produce the hundreds of millions of vaccine doses needed.
6 months. In flu evolution time, that's ages.
It really is. So there's always this risk, this gamble, that by the time the vaccine is ready and people are getting their shots, the circulating virus has already drifted significantly. The match might not be perfect anymore.
And that risk ties right back to what part of the virus the vaccine actually targets, doesn't it? Let's talk about the standard immunological target.
Okay, so traditionally, the vaccine aims to generate antibodies against the hemagglutinin protein, the HA. specifically the globular head region of the HA.
Why the head?
Because antibodies that bind to that head region are really good at neutralizing the virus. They physically block it from latching onto our cells. Stop the attachment, you stop the infection, basically.
Sounds ideal. So what's the catch?
Well, the catch is that HA globular head. It's the single most variable part of the entire virus.
Ah, the best target is also the part that changes the most.
Exactly. It's under constant pressure from our immune systems worldwide, so it evolves rapidly through antigenic drift. This means the powerful neutralizing antibodies we make are often quite narrow. They recognize the specific shape of that season's HA head, and because the head changes, that protection can be short-lived.
Which explains why the vaccine effectiveness, the VE, can be so variable year to year, especially as you mentioned for older adults.
Absolutely. You know, in a good year, with a good match, VE might hit 70, maybe even up to 95% in healthy young adults. But for the elderly, who really need that protection because they're at higher risk for severe disease, VE often struggles to get above 50%. Sometimes it's much lower.
How do we even measure if someone is protected? Is there a standard test?
There is, but it's being questioned.
Yeah.
The main accepted measure, the correlate of protection, is the level of antibodies detected by the hemagglutination inhibition, or HI assay. It measures antibodies that block the virus from clumping red blood cells, which relates to blocking cell attachment.
Okay.
But some of the sources we looked at point out there's actually not a huge amount of solid experimental proof that this single HI measurement tells the whole story.
Wait, so the main metric we use globally might be missing things.
It might be. The concern is that the HI test focuses only on antibodies that block that specific binding site on the HA head, but the virus could be changing in other ways, maybe on other parts of its surface, that affect immunity but aren't picked up well by this test. The sources suggest focusing solely on HI might, and I quote, occlude some forms of antigenic variation. We could be getting an incomplete picture.
Wow. Okay, that really opens things up. If the way we measure change is potentially flawed, maybe the way we think about that change needs a rethink too. This brings us to this fascinating idea, challenging antigenic drift, something called antigenic thrift.
Yes, this is a really interesting alternative perspective. The standard drift model basically assumes the virus is always running away, constantly changing those key spots on the HA head, driven purely by our antibody pressure against those variable bits.
So what's the thrift idea proposed instead?
Thrift suggests it's not just about escape. The hypothesis is that alongside those highly variable spots, there are also regions or epitopes on the virus that have low variability. They don't change much over time.
And these stable spots can still trigger immunity.
Yes. And importantly, they might elicit immunity that provides both clinical protection, meaning less severe illness, and maybe even helps block transmission. So the virus isn't just randomly mutating, its evolution is constrained.
Constrained how?
By this network of cross-protective immunity we all build up from past infections or vaccinations. If too many people have immunity to these less variable conserved spots, it limits the ability of totally new antigenic forms to emerge and spread widely. It sort of corrals the virus's evolution.
That's a fundamentally different way of looking at it. Is there evidence for this thrift idea?
There is, and some of it's quite compelling. Remember the 2009 H1N1 pandemic? Studies afterwards found something fascinating in older people.
What was that?
Many elderly individuals actually had pre-existing antibodies that could recognize and neutralize that new 2009 H1N1 virus.
Wait, how? It was supposed to be novel.
The thinking is these antibodies were likely generated decades earlier, perhaps from exposure to related H1N1 strains that circulated way back, maybe even in the 1970s, like the A New Jersey 76 strain.
So their immune systems remembered a feature, a less variable spot, that was shared between the old strains and the 2009 pandemic strain.
Exactly. It suggests there are conserved epitopes that persist over long periods, and this likely explains why older adults had surprisingly lower rates of severe disease during that pandemic compared to younger adults and children. Their immune memory included these thriftier, conserved targets.
Which is incredibly helpful for vaccine design, right? If those conserved targets exist.
That's the holy grail. Find those stable, conserved regions, and you have a target for a truly broad, maybe even universal influenza vaccine. One that wouldn't need updating every single year.
Okay, let's dive into that full throttle approach then. Where are scientists looking for these conserved targets?
Well, target number one is moving down the HA protein, away from that variable head, and focusing on the HA stalk, also called HA2.
The stalk. Why is that better?
Unlike the head, the stalk region is much, much more conserved across different influenza A subtypes. For instance, the stalk from an H1 virus looks very similar to the stalk from an H2 or H5 virus.
And antibodies against the stalk work.
They do, and they tend to be broadly neutralizing, meaning one antibody can hit many different flu strains. The challenge has always been that the head is immunodominant. Our immune system naturally focuses on the head.
So how do you get it to look at the stalk?
New vaccine strategies are trying clever tricks, like creating headless HA vaccines, basically just the stable stalk protein, or designing nanoparticles that display the stalk in a way that forces the immune system to recognize it.
Interesting. Okay, what's target number 2?
Target 2 is a small part of another protein called M2, specifically the bit that sticks outside the virus, the M2 ectodomain, or M2E.
And M2E is conserved.
Highly conserved across almost all human influenza A viruses. The M2 protein itself forms an ion channel, which is critical for the virus life cycle, so it can't afford to change much.
How do M2E vaccines protect? Is it neutralization like the HA head antibodies?
It's a bit different. M2E antibodies don't typically neutralize the virus directly. Instead, they seem to work mainly through something called antibody-dependent cell-mediated cytotoxicity, or ADCC.
ADCC. What does that mean in simple terms?
It means the antibody acts like a flag. It binds to the M2E on the surface of an infected cell and then signals to other immune cells, like natural killer cells, to come and destroy that infected cell before it releases more virus.
So it's more about cleaning up infected cells rather than blocking initial infection.
Exactly, a different strategy.
And target 3 moves away from antibodies altogether.
Right. Target 3 focuses on cellular immunity, specifically cytotoxic CD8 plus T cells, sometimes called killer T cells.
And what do T cells target?
They don't usually see the surface proteins like HA or NA. Instead, they recognize small fragments of the virus's internal proteins like nucleoprotein MP and matrix 1 M1, which are presented on the surface of infected cells.
And these internal proteins are conserved too.
Very much so. They're essential for the virus structure and replication machinery. So eliciting strong T-cell immunity against NP or M1 could provide really broad protection against many different flu subtypes because those internal parts change very little.
Okay, so we've got these exciting universal vaccine strategies, HA stalk, M2E T cells, but that's longer term. What about improving the vaccines we have now, the seasonal ones?
Yeah, that's crucial too. And there are definitely incremental improvements happening. One big area is moving away from manufacturing the vaccines in chicken eggs.
Why is egg production a problem?
Well, it's been the standard for decades. But when you grow human flu viruses in eggs, they often mutate to adapt to growing better in the egg cells.
And those egg adaptation mutations can sometimes change the HA protein just enough.
That the final vaccine doesn't perfectly match the actual human virus that's circulating. This has been a particular issue with some H3N2 strains, contributing to lower vaccine effectiveness in some years.
So what's the alternative?
Cell-based vaccines, where the virus is grown in cultured mammalian cells, or recombinant vaccines. Recombinant technology, like using insect cells with baculovirus vectors to produce just the HA protein, completely avoids the egg adaptation issue.
Plus, I heard recombinant is faster.
Much faster, potentially. With recombinant, you can basically start production as soon as you have the genetic sequence of the target HA. You don't need to wait to isolate and adapt a live virus strain for egg growth. That can shave valuable weeks or even months off the production timeline.
Which is critical when you're racing against Thrift.
Absolutely. Speed helps bridge that gap.
We also talked about the vaccine focus heavily on HA. Can we improve things just by including other targets in the seasonal shot?
Yes, definitely. There's growing evidence that antibodies against the other main surface protein, neuraminidase or NA, are important too.
How does NA immunity help?
NA antibodies don't usually block infection like HA antibodies, but they seem to limit the spread of the virus in the body and reduce the severity of illness. They're seen as an independent correlate of protection.
But NA isn't standardized in current vaccines.
Generally not. The amount of NA protein in inactivated vaccines can vary a lot between manufacturers and even between batches. It's often not even measured consistently.
So simply ensuring a standardized, effective dose of NA in every seasonal vaccine could give an extra layer of protection.
That's the idea, and it's gaining traction. Another thing is using adjuvants.
Additives that boost the immune response.
Exactly. Things like oil and water emulsions, you might see names like ASO3, AF59, AF03 are already used in some flu vaccines, particularly for older adults. They help generate a stronger and sometimes broader immune response to the standard vaccine antigens.
Okay, so listener, you're seeing these two parallel paths, aren't you?
Mm.
There's the moonshot, the quest for that universal one-shot-for-years vaccine targeting conserved bits like the HA stalk or internal proteins. And then there's the immediate vital work of making today's seasonal vaccine better through faster, cleaner production like recombinant tech by adding targets like NA and using boosters like adjuvants.
And underpinning all this is the virus's global journey. It's not the same everywhere. AH3N2 tends to spread rapidly out from hotspots, particularly in East and Southeast Asia and India.
All the others are different.
Yeah, AH1N1 and the influenza B viruses often show more complex patterns. They can persist locally for longer periods, leading to multiple different lineages co-circulating around the world. It makes prediction even harder.
Which brings us to a really important point, the priority. While the universal vaccine dream is exciting, we can't let it overshadow the urgent need to improve vaccines for the people most vulnerable now.
Absolutely. The elderly, in particular, they suffer the most from severe flu, and they get the least benefit from our current vaccines. Improving seasonal vaccine effectiveness just for that group would be a massive public health victory in itself.
It really would. And that leads us to a final thought for you, the listener, to chew on. We know the hit-or-miss effectiveness of current flu shots sometimes fuels public skepticism about vaccines in general. But we're making huge strides in understanding why it's hit-or-miss mapping those specific amino acid changes on H3N2, identifying targets like the HA stalk. So the question is, which breakthrough will arrive first to really change the game and rebuild that trust? Will it be the technology, like a super-fast manufacturing platform that delivers a perfectly matched vaccine every year? Or will it be the biology, cracking the code for a single-shot, broadly protective vaccine that targets a conserved weakness of the virus? Something to watch.